Okay, hello everyone. I think we'll make a start. So today I just wanted to walk you through our pre-site and talk to you about some of the new species we have on there with a focus on the new human assembly. Okay, so we have new and pre, the new human assembly, GRCh38. We also have Amazon molly, crab-eating macaque, and the hedgehog. So this was of some excitement to different user communities, and some of these were anticipated for a while. So I think some of you, or maybe most of you, do use pre, but if you don't, that's fine. I just wanted to talk a little bit about what it is. So we've got pre.ensemble.org if you want to find it. And one of the big reasons we have it is quality gene sets and annotation take time. So from the time we get the genome assembly, it could be maybe 6 months of work to annotate a high quality gene set that's stable and to do all the other annotation like variation, whole genome alignments, things like that. So we want to make sure that our users can access the genomes kind of closer in a timely fashion to when they come out. So there's preliminary annotation on pre. The genome is there. You can blast against it and you can upload your own data against it and visualize. So most of the pre-species do move to the main site once the full gene builds and once the full release cycle has been completed. So we do have a bit of a restriction on what genomes we take to annotate an ensemble. So they have to be publicly available, high quality, accepted by the INSDC. So that's a big one. And they can be submitted through ENA, DDBJ, or NCBI. So they're all in agreement about the standards and things that they'll take. So, if you do have a genome assembly, do make sure that you submit it through one of those. To the INSDC. So that also means that all of our genomes should be fairly stable and also should be used by different bioinformatics resources because we're all agreeing to use kind of those same ones. So going a little into GRCh38, it's the newest human assembly, and we've chosen to use Venus de Milo to represent this in Ensembl because GRCh37 was represented by David. So we've got 24 chromosomes, so that's good, 1 to 22, the X and the Y, 261 haplotypes, so a lot more haplotypes than we had in the previous assembly, and centromeres for the first time, so those are about 2% of the new assembly. You can find out more about this genome on the GRC blog, so the link is there if you want it. And what has Ensembl done with this assembly? Okay, so we've got repeat findings. So with repeat masker and dust, we have over 50% of the genome has been shown to be repeats with those programs. We do a gen scan prediction, and probably as most of you know, gen scan does over predict, but it is a good way to quickly identify possible open reading frames. So there are 50,000 of those identified. And then we have several gene sets. So I'll talk a little bit about these gene sets. We have the Ensembl preliminary transcript set. So over 100,000 of those. And they're actually shown by default if you do go to a region for the human on pre. I'll talk about that in the next slide, but some people might want to use those, and then others may want to use our mapped gen code 19 set, which is kind of the high quality gen code set that was of human genes on GRCh37. But those have been mapped over, so positions have been mapped over from the previous assembly, our release E75. We also have a RefSeq gene set mapped over from GRCh37 and consensus coding sequences if you want to use those. So just to talk a little bit more about the difference between the preliminary gene set and gen code 19. So both, as I mentioned, are on by default. If you're not sure how to get this view, just go to this tiny URL. So that's the region in detail for the VNN2 gene. And the preliminary gene set is on the way to being a full gene build. So these are the initial alignments, cDNA and protein alignments from the Ensembl automated pipeline. So the good thing about it is everything is mapped straight or aligned, actually sequence aligned, to the new assembly. And also we're using the latest evidence, so the newest proteins in UniProt and NCBI RefSeq, and the newest cDNA. So you'll get, the current picture of the isoforms. So the negative is that because it hasn't not yet been quality checked, for example, you're going to have a lot of redundant transcripts there. They're not stable. We don't have stable IDs on there. And Havana manual annotation, which is clearly high quality, it's manual annotation, has not yet been merged in. So it is very much a preliminary gene set. GenCode 19 does have Havana manual annotation. And when we do have a transcript, which is the same between Ensembl automatic annotation and Havana manual annotation, we'll color it gold. So those are high quality gold transcripts. So you will see those in the GenCode 19 set, whereas we have just the red, automatic pipeline transcripts there in the preliminary set. So this is positive because, we've got Havana manual annotation and it's stable IDs. However, it's been mapped. So if anything went wrong with the mapping between the two assemblies, we may not have a transcript there, or we may actually not have the gene, or we may have a gene in the wrong position. So do keep that in mind. Of course, when we have GRCh38 in the main site, we will have a GenCode 20 probably for human. So if you want to turn on a new, another gene set, for example, RefSeq import, you can click on that cogwheel icon and select it in the configure this page menu. Okay, so that was a bit about the genes. We also have some preliminary variation for you. So again, we've done the mapping from the previous assembly, and we have actually mapped nearly 99% of the short variants. So you can see that 67 million variants. And we've included the 1,000 genome sets. And we also have the variant consequences as well on the Ensembl genes, but also on RefSeq genes, because we do have some users who are specifically using the RefSeq gene set. And you can see here in the diagram, the sequence variants are drawn as vertical lines. So that may be how you're used to seeing them. So you can actually see, once the next slide comes up, the variants in a slightly different way. You can see them with the actual RS or whatever variant ID shown. So I'll show you how to do that in a minute. So this is just to show you that same region in detail for VNN2. I've turned on the 1,000 genomes African population sets and the common 1,000 genomes African population track, which is with population frequencies over 1%, ends up in the common track. And to do that, again, click on the cogwheel icon, and then you have the ability to choose the 1,000 genome set that you want. And then you can actually choose expanded with name to see this kind of track style where you actually have the name of the variant shown. And that will only appear when you're under 10 kilobases. Otherwise, it would just be a mess. And then you have the legend underneath, which shows you the consequence type. So the variants are colored by the consequence type there. So we do have quite a few blog posts already on the new human assembly, and some of this is part of our release cycle. So for example, you could read about the pre-sites. So I've gone over most of what's in that blog post, but there are more statistics in the blog post if you want to see. Assembly mapping, if you want to know more about how that was done from GRCh37, you can have a look at that post. And then we have kind of more personal, although there's a lot of statistics in that one too, a gene builder's perspective on the new human genome assembly. So, focusing on the gene set there. And if you do keep an eye on our blog, we have this GRCh38 category. So keep your eye, there'll be more posts. And so that's everything in pre for human, but what's next then? Well, it will eventually go into the main site, and we're aiming for summer, so it's not too far away, where there will be a full gene build set, so the full GenCode set with the Havana manual annotation in there and stable IDs, whole genome alignments, as well too. We have over 70 species in Ensembl, so it will be involved in pairwise alignments between two different genomes and also the multi-species alignments. Homology and protein families will be determined. VEP access, so our most popular tool, is the variant effect predictor. So that will be available for the new human assembly in the summer, and variation mapping, and also regulatory features. So where the promoters and enhancers, things like that. So that's all I had to say about human, but we do have more species launched on pre. So we have the Amazon molly, and you can see the name of the assembly there, crab-eating macaque as well, and hedgehog. So just a little bit about those. So if you go to pre-ensembl.org, you can see which are the new species by a little new next to it. You can see immediately which assembly is being used, is being launched, and then you can see there are 19 at the moment. And if you click on any of these, like the molly, you actually get taken, and this is the same in our live site as well, you get taken to a species index page or a species homepage where you can get a little information about, for example, the genome assembly, which genome assembly is being used. In this case, the Amazon molly, the genome assembly was produced by the Aquatic Genome Models Consortium. So you can learn a little bit about the assembly there and click through to an example region, for example. It also shows you how to get the molly data via the FTP site or display your data in pre as well. And you'll find out a little bit more about gene annotation. So what kind of things you can find, the cDNA and protein sequence alignments, for example, and some preliminary statistics. So for the molly, we actually have something special. We've got different gene projections from different species. So we have the human Ensembl genes projected onto Amazon molly, and then the stickleback and the zebrafish. So you can get a feeling for what genes other species have in some fish species, in addition to ESTs aligned, cDNAs, and UniProt sequences aligned. So how can you access this type of information on pre? So we've talked a little bit, we talked a lot about the preliminary genes, talked a little bit about the genome sequences there. You can use BLAST or BLAT on any pre-species. You'll see cDNA and protein alignments on any of the pre-species, and you can upload your data as well to visualize it. And you also do have, in addition to the browser, the FTP site there. So that's pretty much all I have to say about the pre-site. As always, send us questions and comments to helpdesk@ensembl.org. You'll see Emily and Denise respond mostly to those, but I respond to some of them as well. So you'll hear from the team within 48 hours. We've got a YouTube channel, a mailing list, courses online, tutorials page, and you can follow us on Twitter, especially is active. Today we got 2,500 participants on Twitter or followers on Twitter, and we've got the blog as well. So just to acknowledge, it's a great team of people, and also acknowledging our funders.